biodegradable. So what does it actually mean to be biodegradable? You sort of stop and think like, well, it means it sort of breaks down into natural products. That doesn't really what you get at the whole piece of it. So the process of biodegradation is really the breakdown of organic matter into its fundamental form. And in a sense, given enough time, everything is biodegradable. Plastics will biodegrade over billions of years when they go back into the earth, come into contact with incredible high rates of pressure, get converted back into oil. So biodegradable in a sense is not a particularly informative word unless there's some certain meaning is brought to it. So a lot of, I think in California, something might be called biodegradable unless it breaks down into carbon dioxide and water. Without being in landfill, it breaks down into your drought. So we're familiar with this process of biodegradation. But let's look at this sort of in the context of ecosystem processes. So when we were looking at the movement of energy through ecosystems, we focused largely on net primary productivity. Secondary productivity as that net primary productivity moved to herbivores, and then productivity as energy is lost moves up to carnivores, and then ultimately to apex predators. At the time, we ignored the possibility that energy could come from something other than that single pathway of primary producer, primary consumer, secondary consumer. And in fact, there's this whole other pathway, sometimes called a detrital pathway, or decomposed organic matter pathway, that is sort of another pathway by which net primary productivity can provide energy to move through food webs. So basically the idea is after you have plant material basically being created via net primary productivity, those leaves might die and senesce. Like often do this time of year, fall down to the ground. And then what happens is they ultimately become this thing called organic matter. And they break down from a leaf into little tiny bits of leaves into ultimately into soil through a process called decomposition, which we're going to today. But that organic matter then becomes ultimately a food source for things like bacteria and fungi. And then these two primary consumers of organic matter might go on to feed arthropods like this mite here, or protozoans, amoeba, flagellates, other sort of soil creatures, which in turn might feed nematodes, or here we have a larger predatory mite, which in turn would feed animals like our mole here, or I suppose birds would feed some of the larger mites, but more often we see salamanders eating some of these soil microbes, or soil microorganisms, macroorganisms. So decomposed organic matter basically provides, it's another pathway by which net primary productivity can enter an ecosystem and provide an energy source for a range of organisms. And it's not the one we think of, it's sure a caterpillar can eat a leaf, but in fact, most of the energy that's moving through terrestrial ecosystems is actually moving through this organic matter pathway, not through caterpillars and birds eating caterpillars. So decomposition results in the movement of energy, but also nutrients through food web. It's a very important component of ecosystems that's often downplayed or So if you're going to analyze or study decomposition, sort of your fundamental equation for doing so is going to be calculating this decomposition rate, K. And K is fundamentally, it's a rate of decomposition. It's going to be tied to some sort of time period. And in terms of the order of operations, relatively straightforward. What we do is we take the data from initial mass, so we might weigh a dead leaf prior to decomposition, weigh it after some set period of time, 6 months, a year, get sort of its final weight, take the natural log of that ratio, and divide that through by time. Now this ratio, because it's going to be less than 1, if you did your math right, it's always going to be less than 1. The natural log of a number less than 1 is always going to give you a negative number. So we throw a negative out here onto the equation to sort of indicate that the rate is going to be positive. So negatives. And so k is going to range anywhere from 0 on up to infinity, but usually it's going to be less than 1. So not all organic matter decomposes at the same rate as we've seen. So if you have two different species of leaf material, if you have oak leaves and maple leaves, sort of in the same physical environment, one is going to decompose faster than the other. And so as a result, we would say one would have a higher decomposition rate measured by K than the other one. So we're actually able to measure those differences. Alternatively, we might take the same species, we might take maple leaves and have them out in the forest, and take those same maple leaves and put them in a decomposition bag and put them in a stream. And those decomposition rates are going to be different because of those different environmental substrates. There are different arthropods and other types of organisms acting on a leaf pad in a stream versus one in the forest. And as a result, the species, the same species, can decompose at different rates in different environments based on differences in temperature, based on differences in the organisms present that are sort of acting at the rate. So K is sort of our touchstone. It's what we can come back to all the time in terms of saying how fast or how slowly something goes. Right. So let's take a quick shot at it. We have some nice sugar maple leaves here. In spring, they look nice and green, and by this time of year, they look kind of sad. They've senesced, they've fallen off the tree, they've started to go through the process of decomposition. Here's some data on those sugar maple leaves. What is the decomposition rate? .76. All right, how did you get that? 2.2 divided by 10. Okay. Okay. Good job. Never forget the negative. And you ended up with 0.76. So that is basically going to give us the decomposition rate on a per year basis. We can see, as you sort of go through the literature on decomposition rates, sometimes the time is reported in terms of days, sometimes in terms of years, sometimes in terms of months, very seldom in terms of decades, because research projects are funded on that. So pay attention to the units, because you might have a situation where you have that same sugar maple decomposing at the rate of 0.02 as a constant, but on a per week basis, right? And it doesn't mean that it's decomposing any faster or any slower. It's the same data, just sort of represented differently. Yeah.
How would you? Is it just 0.76? What per year?
It'll be 0.76 per year in this case. Yes. Yeah, if grams per cent. So think of because it's a decomposition rate, it's going to be some sort of change per unit time. Okay? And again, we don't need units beyond that. All right. This problem's a little bit different, but kind of not. Yeah, 0.015. 0.015. How would we even get that since we don't have any data on mass? Are you sure? I just did it the same way. You did it the same way, but tell me what you did. 32 over 105 divided by going to be 0.015 on a per year basis. Okay. So in this particular case, we didn't have information on the actual mass, but we had something that probably is going to be pretty correlated with mass, basically with volume. It's the same bit of wood, so the underlying chemistry of the wood is not going to change dramatically over time. So from the fact that we've gone from 105 cubic meters down to 32 cubic meters, that'll give us at least a rough approximation of a decomposition rate.
Wouldn't that not be necessarily, as you slow like trying to create trees that per se
Well, because different trees have different densities, there's that's just going to reflect a higher mass per unit volume.
Start over.
Yeah, if we're looking at the case of volume from volume.
yeah, so if you're looking at volume from volume, normally you look at mass or grams per gram.
Right.
So would that be, maybe not as valuable, but it's the right term, but fundamentally different than when you compare it to grams per gram.
I would say not fundamentally different. I'd say it's an approximation, given that we don't have information on grams and grams. Volume to volume is going to give an approximation because, again, the density of the wood is not fundamentally changing within a single species. That's what we can do. If you can come up with a better way of doing a decomposition rate with no additional data beyond this, would be my guess. But if you've ever tried to weigh 105 cubic meters of a dead log, it's kind of hard. But grad students, they can't do it. You gotta get to the gym, yes.
Also going to comment that we use a natural lawn, we not really take so long.
You can, just, you know, I've been waiting for years for someone to do that, and you're that guy. I'll let you stay there. All right, any questions on calculating decay rates or decomposition rates? Quite simply, it's solving for K, and the higher the value for K, the more rapid the decomposition. The lower the value for K, the more slow the decomposition. And because our log and our sugar maple leaf, we're sort of also both looking at K on a per year basis, 0.76 versus a 0.015. So the log is decomposing at a more slowly than our sugar maple leaf, which hopefully makes intuitive sense. I want to take sort of a brief detour into some biochemistry. Don't worry, not that kind of biochemistry. In terms of looking at what's decomposing during the process of decomposition, we can look at decomposition of leaves. And there's basically three fundamental components of a leaf that undergo decomposition. We have cellulose, we have hemicellulose, and we have lignin. And sort of the thing to keep in mind is cellulose and hemicellulose are basically sugars that are easily digested. Whereas lignin is an awful molecule that is really, really hard to digest. It requires a high degree of energy input to break down lignin. While there are enzymes that can break down lignin, most of the time lignin breakdown depends on oxidation. And so it ends up being sort of a very slow process. However, rather than listen to me talk about breakdown of cellulose.
Understanding plant litter decomposition is a key part of understanding the amount of carbon in the soil and its role in global warming. In order to appreciate the process of plant litter decomposition, it's important to understand the anatomy of a leaf. Leaves are made of many specialized cells, which determine its shape, size, and function. Surrounding each cell is a cell wall, and between each of the cells is an interstitial space. The cell wall surrounds the cytoplasm and intracellular components, most of which are leached out shortly after leaf fall. The cell wall supports the overall leaf structure, protects the cell, and separates the intracellular components from the extracellular environment. The inside layer is the plasma membrane. In between these two layers is the primary cell wall, which is the main target of decomposition. The three main components of the primary cell wall are cellulose, hemicellulose, and lignin. Cellulose consists of sugars known as glucose. Each individual sugar molecule is linked together to form a long linear polymer. These long linear polymers group together to form a crystalline structure. Since cellulose consists of sugars, it's a high-energy food that microbes attack first in decomposition. Hemicellulose is a polymer made up of chains of sugar molecules, primarily xylose and arabinose. These are also high-energy foods. Hemicellulose consists of shorter chains and has a branch structure. Hemicellulose encases cellulose to form a microfiber and plays an important role in holding everything together. Lignin is a complex compound made of different kinds of phenol-based molecules, which are arranged in random patterns to form large polymers. Lignin traverses the spaces between cellulose bundles. It's a low-energy food, which requires a lot of work to break down and shields the high-energy foods cellulose and hemicellulose. Microbes are microscopic organisms that belong to groups such as fungi and bacteria. They are the most numerous organisms on Earth and are sometimes referred to as germs. They can live everywhere, from air, soil, vegetation, and water to the human body. Microbes play an important role in almost everything, including leaf decomposition. During decomposition, microbes break through the leaf cuticle and traverse the interstitial space on their way to the cell wall. Microbes find food by releasing a small number of enzymes that break down complex compounds, like cellulose, into simpler ones, such as glucose, that can then be consumed. An increase in the concentration of these simple compounds tells the microbes to produce more of the needed enzymes for more release. The three main structural components of a leaf that microbes break down are cellulose, hemicellulose, and lignin. Microbes attack these high-energy foods, such as cellulose and hemicellulose, first. Lignin is a low-energy food, which shields cellulose and hemicellulose. Microbes only attack lignin as a last resort to get the protected high-energy food. There are two methods enzymes use to break down large, complex compounds into simpler, smaller ones that they can eat. One is the lock and key method, which is used to break down cellulose and hemicellulose molecules, which are chains of sugars, into single sugars that can be consumed. In this process, the enzyme is the key. The key fits the lock on the matching substrate molecule and causes a break in the chain. Some enzymes use this method to break a long chain in the middle to create shorter chains. Other enzymes clip off the ends to release individual sugars. Typically, multiple enzymes are packaged together into a complex scaffold. The scaffold organizes the enzymes that break bonds on both the ends and the center of the chain. To completely break down the chain, the scaffold slides up and down the chain and takes it apart. The other way enzymes break down complex compounds is by oxidation. This process is used to break down lignin. Lignin has a random structure. Every molecule is different, so the lock and key method does not work. Also, it takes a lot of energy to break down lignin, which is a very stable molecule. Because of this, enzymes do not have enough energy to attack lignin directly. Instead, enzymes attack other molecules that create free radicals, which are highly reactive compounds. These free radicals then react with the lignin and break it down. Once the lignin is removed in this way, the cellulose and hemicellulose that was encased by the lignin is available for decomposition. As microbes consume these foods, they are releasing carbon dioxide into the atmosphere through the process of respiration. Scientists need to understand how leaves decay and what influences how fast this process occurs so that they can predict how much carbon dioxide will be produced during decomposition.
All right, That's the relatively short version of decomposition of plant material. Some leaves will have relatively large amounts of cellulose and hemicellulose, less lignin. Other ones will have lots and lots of lignin, less cellulose and hemicellulose. And I think if you can remember which ones are high energy and easy to digest, cellulose and hemicellulose, which ones are low energy and hard to digest, lignin, should be in good shape for the exam. Note to self. Exam question. Just snuck right past it. If anyone's ever seen the fungus, turkey tail fungus, it's a little bracket fungi that grows on dead wood, which tends to have some really nice colors that sort of give it its name, turkey tail. It's not very big. It's one of the fungi that is probably most important for breaking down lignin forests. I've seen turkey tail fungus pretty much everywhere from the tropics all the way up to the boreal forest. Generally between, it's very widespread, and probably because it's able to take advantage of a lot of lignin species. So as we continue thinking a little bit more about decomposition, one of the things that's important is not just the chemistry of the substrate that's being decomposed, but also the environment in which decomposition is taking place. And we see decomposition rates can vary quite a bit among various parts of the earth. So I've pulled together sort of 5 biomes here. Tundra, where it's wet but very cold for most of the year, where you actually have a growing season of 2 to 3 months a year. Not a lot of time for temperatures to be above freezing for decomposition to occur. So the tundra, decomposition, pretty slow. Equatorial tropical rainforest, every day, every night, 12 hours in length. They typically get 4, 8, 12, in some places 16 meters of rain a year. We get almost 1 year a day. So lots and lots of rain. Temperatures seldom fall below 90 degrees Fahrenheit, or roughly 20 degrees Celsius. It's hot, it's humid. If you're a microbe in a future life, which you will be, at least parts of you, tropical rainforest is the place to be because decomposition rates are going to be very high in a warm, wet environment. So those sort of represent extremes. And then sort of in between, we have the boreal forest, or taiga, which stretches circumpolar mainly through Canada and through Russia, dominated by conifer trees, again with relatively short growing season, typically about 4 months a year. So frozen for 8 months a year, decomposition rates are going to be higher than we find in the tundra, but still not great in the great scheme of things. And then in the temperate zone, we have things like temperate forests and temperate grasslands. Temperate forests are supported where there's a bit more moisture. You get temperature extremes that fluctuate a bit more in temperate grassland areas. So these are sort of more or less equivalent in terms of decomposition rates that you would expect in these environments, although probably a little slower in temperate grasslands due to the fact that it tends to get a little bit drier, and they do tend to have sort of short periods of drought over the course of the season, which can affect decomposition rates. So what I've done in terms of describing these various biomes and conditions for decomposition, it should provide you a relatively solid base for being able to predict what decomposition rates might be. So if we had data that sort of look like this, where each color represents one of these different biomes, and we're looking at decomposition rates of leaves and of roots over a 10-year period, which color would correspond with which particular biome? So give me one color, one biome. What's the matchup?
Boreal forest and red.
Boreal forest and red. And what's the rationale here?
So the tundra is gonna be in the extremity at the low end. Boreal forest is gonna be close to tundra, in terms of things, so it's gonna be the one above the tundra. So the decomposition rate.
Uh-oh, I'm seeing a lot of hands going up here.
Okay.
Where's the brown one, the tropical forest, if you come to this one?
Oh, yeah, wait, I misread it.
Ah, that's right.
So, yeah, green.
Okay.
Boreal.
Alright, so the green's gonna be boreal, and you've got the tropical rainforest. Alright, so what else is left? Who's gonna take the,
Purple's gonna be tundra.
purple's gonna be tundra, that was an easy one. Alright. Alright, who's gonna rise to the challenge for temperate grasslands and temperate forests? Yeah.
The forest is gonna be the red one.
The forest is gonna be the red one, how come?
It's more rain.
Just a little bit faster. In the forest.
Yeah.
The grassland?
The grasslands, right? Forest had the grasslands blue.
Oh, I don't know. Tough to disentangle, because I mean, like the roots, it's kind of overlapping after 10 years. Leaves, there's some clear segregation. Kind of tough segregating those out.
But I mean, the same can be said about the tundra and the boreal forest.
Well, probably not. I mean, the leaves are sort of matched up, but the roots are decomposing more quickly, and then the roots are not going to decompose very quickly in tundra because the ground's frozen, there's not an opportunity for them.
Yeah, but there's no. But I would still say the temperate, the deciduous forest would still be red because we are seeing a faster decomposition.
I think you're right.
Alright.
Alright.
I think we reached,
We've reached a foundation. Anyway, not knowing what's decomposing, we should be able to use some broad information about climate to roughly figure out rates of decomposition being faster, being slower.
how does the percent remaining, of mass remaining go up at certain points?
You're talking about like from here to here?
Yeah, like groups.
Yeah, that's fundamentally going to be measurement error because when you're doing this, you can't sort of go out and then measure something and then basically put it back because in the process of measuring it, you're usually drying it out and then weighing it back in the lab. So what you would do for a 10-year study is you put out 100 bags, and each year you take 10 bags back with you, and sort of sampling with the same substrate per year. You're getting an estimate. So sort of left off of this graph are the error bars that would represent the variability in each of those measurements. So the fact that you've gone sort of up a little bit in 1 year suggests that for whatever reason, this probably was not a particularly favorable year for decomposition. So it was probably pretty slow, and you didn't actually measure much decomposition that particular year at that site. That's basically why it'll go off in some years. Probably more information than you want to know, but that's how they roll. So in your senior text, you had a chance to sort of look at these changes in leaf mass over time. It sort of starts off, you've got sort of a drop, and then you get this sort of nice exponential decay function. And chances are you haven't given much thought to why is exponential decay, not linear decay. So what I'd like to do is, in terms of looking at litter decomposition through this process, is try to think about it from the perspective of the organisms that are doing the decomposition. So far we've been looking at it the eyes of a scientist, we've been weighing bags, calculating decay rates. Let's look at it sort of from the organism's perspective, because this can actually be useful, and it helps us think about things in a more creative way. So, if you're a microorganism and you spend your life as a microorganism, as a decomposer, you sort of come upon this wonderful resource of leaf. You're like, oh, look at this leaf. Life is grand. And as you basically go and begin to consume that leaf, what are you going to consume first? Okay, the higher energy or the more palatable stuff. So what would that be specifically? Okay, so you're going to hit the cellulose and hemicellulose first. Unless you're completely irrational and like, I like lignin, No. You're going to hit the cellulose and hemicellulose. And so sort of in these very early stages of decomposition, what's being broken down initially is these high energy sugars. So you initially get sort of a very rapid rate of decline in the overall mass of that leaf as those sugars are basically being consumed by those decomposing organisms. However, as the organisms work through the most digestible parts, or the best bits, the decomposition rate is going to slow, because you get to about here, and you're looking around, and you're like, oh, there's some hemicellulose over there, so go over there. There's a little over here, and then eventually you've got sort of this big lump of lignin. And lignin. How to accurately describe lignin. If you've ever eaten a cardboard box, that's basically mostly lignin fibers, with very little cellulose and hemicellulose contained in there. So sort of you get down about here and you're like, man, all that's left is basically the worst bits of this whole thing. And so what ends up happening, again, because this is a low energy food, it takes a lot longer to consume lignin. So the rate at which that leaf mass is being consumed basically begins to slow. So this exponential decay function is largely a consequence of the fact that when that leaf or that substrate is consumed, all the best bits are eaten first, all the worst bits are saved to the very end. So leaf chemistry can be an important predictor of how quickly or how slowly something decomposes. Let's try this. Sort of based on what we know about leaves and leaf chemistry, of these two different genera of plant species, both of which are tropical species, Schizolobium and Hyramonema, which one do you think is going to decompose more quickly? Which one's going to decompose more slowly? How would you know? Let's start with how would you know? What are you looking at for some information?
The slow would decompose faster because it has a lower carbon to nitrogen ratio.
Okay, lower carbon to nitrogen ratio. But what if, let's see, what if this also produces some very powerful alkaloids in those leaves, which allow them to have a lower carbon to nitrogen ratio because it's highly toxic for things.
That's probably the other one.
Then probably the other one. So I think your reasoning is right in terms of thinking about carbon to nitrogen ratios. There's lots and lots of carbon, lots of nitrogen relative to the carbon. So that's going to indicate a more nutritious substrate for the decomposer compared to our other species. What other data on here might be useful? Yeah.
You can also look at the amount of lignin and the amount of cellulose. Just kind of make a nice ratio if you want to. But
Okay, so we've got more lignin, but also more cellulose, and roughly equivalent heavy cellulose too. You have lignin to nitrogen ratios. Yeah, so, 1% nitrogen.
Did you look at the lignin, whichever one had more lignin?
Lignin can also be a good indicator. Again, there's also the problem of what chemistry is not included in the chart, right? So, as scientists, we often find ourselves in situations like this. We haven't figured out all 20,000 chemicals present in a leaf, right? We take leaves back to the lab and we run through a relatively standard set of assays. This is actually a little more complete than most. But we might look at leaf nitrogen and leaf phosphorus. And carbon to nitrogen ratios are often measured in terms of leaves as a quick and ready proxy for figuring out how high quality or low quality the leaf is going to be. These researchers also measured lignin, which most researchers don't do. And we can see that the lignin to nitrogen ratio is much higher in one species than another. It's nearly, not nearly, quite 3 times as high, about 2.5 times as high. So, as scientists, we don't have all the information that we need to make an exact answer. So what we often do is we make predictions. Based on the data that we have and based on what we know, if we had to make a safe prediction, we would say which one's going to decompose faster. Schizolobium. Remember, there's a right way and a wrong way to mispronounce that. Schizolobium, thank you. It's actually true. Schizolobium is more likely to decompose more quickly based on its lower carbon to nitrogen ratio and based on its lower lignin to nitrogen ratio. So what does the data actually say? Sure enough, schizolobium, shown right here in triangles, shows the fastest rates of decomposition, decomposing by nearly 90% in less than a year. There's tropical forest, of course, it's very quick. Hyreema, decomposing or losing not quite half its mass over that same period of time. Now, we can't conclude that that's due strictly to its carbon to nitrogen or its liquid to nitrogen ratios, but it's probably a good guess that that's playing a major role in the rates at which each of these decompose. If you wanted to, you could go back and see how well Cecropia, Inga, and Saba match up with our chart. Not required, but who knows? Maybe an exam question. Saba versus these are all species we will see in the summer forest array. Any questions on the role of climate or the role of substrate or leaf chemistry in terms of affecting rates of decomposition? Yes.
So higher nitrogen levels help break down more, that kind of where you're going with it.
Well, let's think about the nitrogen in context. If you have lots of nitrogen relative to the amount of carbon present,
Yes. Okay.
yes,
Yeah.
with lots of nitrogen relative to the amount of lignin, yes.
Okay.
Yes.
Okay.
But nitrogen in and of itself is usually not going to be a very powerful indicator.
Okay. That's how I meant it.
Because lots of plant poisons are alkaloids, which are nitrogen-based compounds. So just knowing the nitrogen is not always going to be informative. It could be just one big poisonous mass. Right? But in context of carbon,
know what parts of the information out. You said high nitrogen, what other common compounds in all points of the poisons?
There are nitrogen-based poisons, alkaloids, and then a lot of carbon-based poisons in terms of phenolics and tannins. Those are sort of our two major classes of poisons to keep your pores away.
Okay. So it's high in both. It's supposed to be high carbon, right?
Potentially. Just the nitrogen-based compounds tend to be deadly, whereas the carbon-based compounds tend to be safe.
Yeah.
I'd like to finish up returning to the process of disturbance and succession. A couple weeks ago, we were looking at the Yellowstone fires in 1988, post-fire succession. It is now Yellowstone fires in 1988 in California.